So, today we will start our next lecture, that is cardiac muscle physiology. So, in this lecture we will deal with the various topics of structure of a cardiac muscle fiber and mainly how an excitation contraction coupling mechanism occurs in a cardiac muscle fiber and how it is different from a skeletal muscle fiber. In brief, we will study. So, generally the cardiac muscle fiber consists of two types of fibers. The fiber which is mainly responsible for the contractile tissue, that is the atrial and the ventricular musculature, and for the conducting tissue, that is the origin of the cardiac impulse and propagation of the impulse. So, we have contractile tissue, atria and ventricles, we have the excitatory tissue and the conductive tissue, that is the pacemaker tissue or the nodal tissue. So, the two types of cardiac muscle fibers, generally the histology of this cardiac muscle fiber or the physiological mechanisms are the same, except the resting membrane potential which is generated in the nodal tissues and the atria and the ventricular muscle, that is the myocardium. These two are different. One is the ventricular muscle potentials and other one is the pacemaker potentials. So, this we will discuss in further lectures. So, generally the characteristics of cardiac muscle is almost similar to that of a striated skeletal muscle with few differences. The first and foremost is it is involuntary in nature. Our skeletal muscle is voluntary in nature. It also has got striations, it also has got various cell organelles, mainly mitochondria because of the contractile property, since it has to contract. The same is with the cardiac muscle. Now, the main difference is the skeletal muscle fibers are enclosed in a closed loop and each muscle fibers are separated, the muscle bundles are separated by the connective tissue, but that is not the case with the cardiac muscle. Cardiac muscles are branched, but they are interconnected with each other. There is interdigitations with each other, and that is the mainly role and that is mainly required for the functional syncytium mechanism which is seen in the cardiac muscle. Now, the heart, if it has to pump, each muscle fibers can't pump individually according to their own characteristics or nature. It has to pump as a whole. The atria has to pump as a whole, the atria has to contract as a whole, the ventricles has to contract as a whole. This coordinated action in the cardiac muscle fibers, in all the cardiac muscle fibers, along the cardiac muscle fibers is mainly regulated by the syncytial property or the functional syncytial property, which is mainly because of the intercalated disc present. The interconnections between the cardiac muscle fiber is mainly done by the intercalated disc. Now, intercalated disc are usually dark lines which is seen in the light microscope. So, this you can see the intercalated disc. The magnified version is shown over here. The cardiac muscle fiber also consists of the thin and the thick filaments, that is actin filaments and the myosin filaments. It has also got the Z line. The sarcomere also has got the A band, I band. The only difference is the intercalated disc. Now, this is the intercalated disc. Now, this is one cardiac myocyte, this is one cardiac muscle fiber and this is one cardiac muscle fiber. These two cardiac muscle fibers are joined mechanically by desmosomes and electrically by gap junctions. So, mechanically the two muscle fibers are joined by the desmosomes. In the intercalated disc, what happens? The ends of the adjoining cardiac myocytes are usually joined mechanically by the desmosomes and electrically by the gap junctions. Now, mechanically because so that they will contract as a whole, electrically because the electrical impulse should travel across the cell from one cell to another cell, since we know the electrical event precedes the mechanical event. So, the electrical syncytium is mainly maintained by the gap junctions and the mechanical syncytium is mainly maintained by the desmosomes. These are nothing but the intercellular junctions, cell junctions. So, gap junctions mainly act as an electrical synapse over here. So, that's why the cardiac muscle acts as a mechanical as well as electrical syncytium. Now, at this point, the intercalated disc, as I told you, which is an extensive enfolding of the membrane, the gap junctions which provide the electrical syncytium in the intercalated disc, they provide low resistance at intercalated disc. The gap junctions, these are the proteins, the channel proteins which are present, and they are made up of connexin. The protein is connexin. 6 connexins form one connexin. So, this gap junctions are made up of this connexin and 6 connexins. So, these is mainly the electrical impulse, electrical synapse, and the low resistance channel proteins or the low resistance vessels, so that there is no barrier to the flow of the electrical impulse. So, this allows the action potential to travel from one cardiac muscle to the other cardiac muscle. So, as a whole, the cardiac muscle forms two separate syncytium. One is atria, the other one is ventricles. The atrial syncytium is formed and the ventricular syncytium is formed. Now, atrial syncytium and ventricular syncytium is generally separated by the continuous fibrous band, except at one place, that is AV bundle or atrioventricular bundle. Now, each syncytium obeys all or none law. All or none law means either it will contract as a whole or it will never contract. It will not contract at all. It will contract as a whole or it will not contract at all. So, it is not that it will partially be contracted state like our skeletal muscles, the muscle tone is always maintained by the partially contracted muscle. It is not like that. Heart is never like that. Heart, either it will contract as a whole or it will never contract. So, each syncytium obeys all or none law. Now, each muscle fiber is about 100 micrometer and 15 micrometer broad. The cytoplasm which is present in the muscle fiber is sarcoplasm, the same as that of the skeletal muscle fiber, which contains abundant of cell organelles, especially the mitochondria, because the contraction needs lots of ATP, and each muscle fiber is made up of number of parallelly arranged myofibrils, and myofibrils consist of both thin and thick filaments acting as myosin filaments. Now, again the difference comes in the sarcotubular system. Now, the tubular system in a skeletal muscle, the sarcotubular system is usually the formation of triad. The triad is formed in the skeletal muscle. There is a T-tubule. This T-tubule is an invagination of the sarcolemma into the deep into the cell, and there are two cisternal end in case of triad, but this is not the case with that of cardiac muscle. In cardiac muscle we do not get triad, but we get dyad. The another important thing is the T-tubule penetrates sarcomere at the level of Z line, but in case of skeletal muscle this penetration occurs at the junction of the A and I band. So, why there is difference of this formation of triad and dyad? Now, as you can see in this diagram, this is the triad of the skeletal muscle, the pink one is the T-tubule, this two light green color, these are the cisternae. So, there are two cisternae and one T-tubule forming a triad structure in a skeletal muscle. This T-tubule is very lean while the cisternae is very much developed. The cisternae is the sarcoplasmic reticulum, the storehouse of calcium. In case of dyad, which is seen in the cardiac muscle, the T-tubule is very much more in case of diameter. It is almost 5 times, the diameter of the T-tubule is 5 times than that of the skeletal muscle, which is present in the cardiac muscle, as you can see. And the cisternae, that is the sarcoplasmic reticulum of the cisternae of the sarcoplasmic reticulum, that is not well developed as that in the skeletal muscle. Also, this cardiac muscle T-tubules consist of some mucopolysaccharides. So, because of this few reasons, They exhibit dyad in the cardiac muscle and triad in the skeletal muscles. Now, if we see the further detailed molecular structure, this is the transverse tubule, this is the invagination of the T tubule, invagination of the sarcolemma deep into the cell. This is the cisternae, that is the sarcoplasmic reticulum. Now, this is the T tubule, this is the sarcoplasmic reticulum. The channel proteins which are present on the surface of the T tubules, that is L type of calcium channels, LTCC, this is L type of calcium channels. Since we have already discussed what are the various types of calcium channels, so this is L type of calcium channels, also known as DHPR, dihydropyridine receptors. Now, the second channel protein which is present in the cisternal surface of the sarcoplasmic reticulum, this is RYR2, this is nothing but the ryanodine receptors. This is the ryanodine receptors. So, we have channel proteins. These two channel proteins are very specific, like T tubules bear the L type of calcium channels, and the cisternal end of the sarcoplasmic reticulum, they bear the ryanodine receptors. And the connection between there is a connecting link between the T tubule as well as the cisternae of the sarcoplasmic reticulum, that is mainly done by the junctophyllin 2 protein. This is a channel which usually binds the transverse tubule and the sarcoplasmic reticulum of the cisternae. So, with this we will move on to the excitation contraction coupling mechanism in a cardiac muscle. The initially what I had already told, that electrical event precedes the mechanical event. Since cardiac muscle is an excitable tissue, the property of excitability is present in the cardiac muscle. So, there is always an electrical event which will occur first. That means, there will be generation of action potential through depolarization mechanism that will occur first, and that will follow the contractile mechanisms or the mechanical phenomenon. Now, resting membrane potential of a normal cardiac muscle is minus 85 millivolt to this minus 90 millivolt. This is specifically when we are talking about the ventricular myocardium or the muscle potential. The pacemaker potential is around minus 60 millivolt, that is different. So, what we can see, most of the thing which has been already discussed, suppose this is the T tubule and this is the cisternal end of the sarcoplasmic reticulum. So, this is the sarcoplasmic reticulum and this is the T tubule, the invagination of the sarcolemma. It is bearing certain channels, the very important channels, that is L type of calcium channels, and the cisternal end of the sarcoplasmic reticulum, they are bearing the channels, that is ryanodine receptors. So, this is the L channels and these are the ryanodine receptors. Now, whenever there is influx of action potential, whenever there is action potential generated and it traverses the T tubule, these channels are the voltage sensors. Voltage sensors means there is more positive charges over here. So, this more positive charges will cause this L type of calcium channels or other known as DHPR, which are nothing but the voltage gated calcium channels. Voltage gated calcium channels. So, this voltage gated calcium channels will open and there will be influx of calcium. Now, influx of calcium is occurring from extracellular to inside the T tubules. So, extracellular calcium triggers the L type of calcium channels. So, this extracellular calcium triggers or enters through the L type of calcium channels, the voltage gated calcium channels will open and the calcium will get influxed from extracellularly inside the this T tubules, and obviously it will stimulate the other receptors, that is ryanodine receptors. Now, these ryanodine receptors are present where? These are present in the cisternal end. Now, the T tubules and the cisternal end are very close to each other, though there is a gap which has been shown in the figure, but it is present practically it is present very close to each other. So, they will also sense the voltage since the calcium is also entering, because there is more positivity. This ryanodine receptors, what will happen, this ryanodine receptors, this will also cause release of calcium from sarcoplasmic reticulum to the sarcoplasm, that means to the cytoplasm. So, there will be calcium which is released from sarcoplasmic reticulum to this sarcoplasm. Now, this calcium which is released, this will bind to the filaments which are present in the sarcomere, that is we have the thin filaments and the thick filaments, and this calciums will bind and will cause the cross bridges formations, the same which is occurring in the skeletal muscles also, and there will be contraction of the muscle. So, this is usually the excitation contraction coupling mechanism. Now, the main difference of this with the skeletal muscle is, in the skeletal muscles there is only the endoplasmic calcium which is mainly required for the contraction, but in case of cardiac muscle it is calcium induced calcium release. That means, extracellular calcium is also present as well as sarcoplasmic reticulum calcium is also present. Now, this is the diagram which we can see, this is the T tubule and this is the sarcoplasmic reticulum which has already been told. Now, this is the L type of calcium channels and here we have the ryanodine receptors. Now, whenever these are the other pumps which is mainly responsible for the muscle relaxation. Now, whenever there is generation of action potentials, whenever this action potential comes, we can see because of this action potential there is calcium ion which is entering through this L type of calcium channels, and that will trigger calcium release from the sarcoplasmic reticulum with the help of ryanodine receptors. So, more amount of calcium gets released in the sarcoplasmic reticulum. So, the sequence of events is action potential moves along the T tubule, then we have activation of the dihydropyridine receptors or the L type of calcium channels, that is nothing but the voltage sensors which senses the electropositivity, and calcium then binds with the ryanodine receptors which opens or releases large amount of calcium, and this large amount of calcium is nothing but we call as calcium spark. So, this is nothing but calcium induced calcium release, CICR. So, calcium induced calcium release that results in calcium sparks. So, calcium from extracellular fluid contributes in contraction. So, this is mainly through the ryanodine receptors which is present at the sarcoplasmic reticulum. Now, how the muscle relaxation is exhibited? Now, already we had seen that in case of skeletal muscles also the same steps occur in the muscle relaxation, that is the cross bridge linkages, they detach themselves, there is ATP synthesis, and finally calcium is thrown outside. Now, calcium is thrown outside with the help of two mechanisms. The first one is sodium calcium exchanger and the other one is calcium pump, that is Sarka pump. Now, there has to be two pumps because the calcium which is already entering into the sarcoplasm during the muscle relaxation, that should enter into the sarcoplasmic reticulum. So, this Sarka pump will take the calcium back from the sarcoplasm or the cytoplasm to sarcoplasmic reticulum. Now, usually this Sarka pump is kept inhibited. The name of Sarka pump comes from sarcoplasmic or sarcoendoplasmic reticulum calcium ATPase pump. So, this is usually kept inhibited by a molecule known as phospholamban. So, this phospholamban is the molecule which usually keeps inhibited, which usually inhibits the Sarka pump. This inhibition will removed only whenever there is a phosphorylation of this molecule, and this phosphorylation is done by none other than cyclic AMP mediated protein kinase activity. Cyclic AMP mediated protein kinase activity. Generally, there is a very common drug, important drug known as epinephrine or adrenaline. They usually cause or affect this Sarka pump activity, or adrenaline or epinephrine. So, Sarka pump, which is usually kept inhibited by the phospholamban molecule. This phospholamban molecule is usually the inhibition is wiped off whenever there is a phosphorylation by cyclic AMP, which is protein kinase activity mediated, and this is usually done in case of drugs by the adrenaline or the epinephrine. Now, the second thing is, whenever the calcium is going inside the Sarka pump, the calcium should not come out of the sarcoplasmic reticulum. So, there should be a calcium binding protein in the sarcoplasmic reticulum. So, what are this calcium binding protein? The calcium binding protein, mainly we have two calcium binding protein. The one is calciquetrine, the other one is calreticulin. This two we have to remember. The calciquetrine is very important. It is found in most of the muscles, the skeletal muscles, cardiac muscles, but most importantly calreticulin is present in smooth muscle, is abundantly present in the smooth muscle, calreticulin. So, these are the calcium binding proteins. Whenever calcium will come to the Sarka pump, this calcium binding proteins will bind to the calcium, and this calcium will not be able to then move out of the pump. So, this is the role played by the calcium binding proteins. Now, back into the T tubule also you need to throw out the calcium. So, that is usually done by the sodium calcium exchanger. So, sodium calcium exchanger molecule is there, that usually one calcium is exported for import of 3 sodium ions. 3 sodium ions will come inside and 1 calcium ion will be thrown out. So, this portion had already been told, that is sarcoplasmic reticulum calcium pump is inhibited by the regulatory protein phospholamban, and when phospholamban is phosphorylated by cyclic AMP dependent protein kinase activity, this inhibition of the Sarka pump is lost, and this is mainly attributed by the drugs like adrenaline and epinephrine if you want to increase the contractility of the heart, so, and decrease the relaxation rate of the cardiac muscle. So, in this way muscle relaxation occurs. So, this is the main difference which is seen in the skeletal muscle contraction as well as the cardiac muscle contraction in case of muscle relaxation. The coupling comparison overall, the trigger for the sarcoplasmic reticulum release is mainly voltage dependent in case of skeletal muscle. In case of cardiac muscle, the trigger is calcium induced calcium release. So, it is the very important question which is asked, which is the main influencing factor, that is the extracellular calcium which enters the T tubule and further causes release of the calcium from the sarcoplasmic reticulum, and the calcium release is proportional to the membrane voltage in case of skeletal muscle, while in cardiac muscle the calcium release is proportional to the calcium entry. It is not that proportional to the membrane voltage, how much calcium is entering. So, the calcium release will also occur in that way, that is why we get calcium sparks over there. So, the contractile phenomenon is seen in the cardiac muscle and the skeletal muscles. Mainly in case of skeletal muscles, the applied aspects generally we will talk about related to the calcium voltage gated channels, that is malignant hyperthermia, that has already been discussed with the calcium voltage gated channels, but that does not occur in case of cardiac muscle because of the calcium induced calcium release. So, that is why in this lecture we conclude by discussing the structure of the cardiac muscle, what is the difference between triad and dyad in the skeletal muscle as well as the cardiac muscle, how the excitation contraction mechanism distinguishes between the cardiac muscle from the skeletal muscle. So, with this I would like to conclude today's lecture. Thank you.